Welcome everybody in today's Cell Talk presentations. Today's Cell Talk will be presented by Dr. Vijay Parul. Vijay is a senior scientist at Renix who leads bioinformatics, genomics, and next generation sequencing research related activities in the organization. He is currently working on the neoantigen cancer vaccine pipeline projects here. Vijay holds a PhD in life sciences from TMC Actrec, Homi Bhabha National Institute, Navi Mumbai. His PhD study focused on cancer genomics. Previously, Vijay has worked as assistant professor at graduate and postgraduate degree colleges as well. At Renix, he has gained experience in scientific, operational, and administrative matters. Okay. I welcome Vijay for his presentation today.
Thank you, Dr. Nupur, for the introduction. Today, for our routine Cell Talk Journal Club activity, I have selected a paper whose title is Sex and Age-Dependent Genetics of Longevity in a Heterogeneous Mouse Population. The original authors are Maren Baus-Levan et al. The title for today's presentation is A Multicenter Study Uncovers Genetic Factors That May Play Significant Role in Mice Lifespan. The broader topic selected is longevity. This paper was published in journal Science, which has a 5-year impact factor of 54.5. This paper was published in year 2022, and the research presented in this paper has an impact on aging and longevity. So, introduction. We are all interested to know the aging process and the longevity. Everybody wants to live a long life. But as for now, even science hasn't fully understood the process, how this aging process is governed, and how much a particular organism will live. So this is not yet fully understood process, and here molecular, cellular, and organismal homeostasis, it is affected by multiple environmental and genetic factors. So two major factors are here, which are environmental and genetic factors. Most of the environmental factors have been studied, such as pollution, nutrition, etc. But genetic factors, these are still a mystery. Which of the genes or genetic locations in a human genome determine the aging process and the longevity? This is the topic of interest for current science, and this is a area where this paper is focusing on. So aging process, it is a process which results in the progressive functional decline in physical, mental, and reproductive capacities of the organism. In younger age, the organism is developing, and the rate of growth is more, and the rate of growth is compensated by the rate of degeneration. As the person ages to the middle ages, and that rate stabilizes, and the person goes into the latter ages, there the degenerative processes are more as compared to the regenerative processes, and therefore it leads to functional decline in physical, mental, and reproductive capacities of the organism, the molecular processes of which are not fully determined. Aging process, it leads to affliction of multiple morbidities and consequent mortality of the organisms. And current human population, this is a major challenge, because as the population of Earth is increasing, and also country is also increasing. So in future, there will be a huge population of aging people who will require medical aid and life care in their latter ages. So this is very much essential that we know what are the processes that govern the aging process. Understanding of the environmental and genetic factors that are central to the aging is very important in that respect. Coming to the general background, aging process affects molecular, cellular, and organismal homeostasis and leads to functional decline in the organism. So variety of processes, various processes that are involved in the homeostasis of the organism to maintain its shape and functionality, shape, size, and functionality. These are involved in the aging processes which are getting affected. The aging process is affected by multiple environmental and genetic factors. And in this particular research article, we are focusing on both environmental and genetic factors as to specifically isolate certain genetic factors which points to this aging process. Genetic studies have identified many of the conserved genes and mechanisms affecting aging, but still we have not pointed out any specific region which are which are we are sure that they are involved in the aging process. From the metabolic perspective, the factors that control aging are affected by longevity. These include nutrient sensing pathways, which are the growth hormone and insulin-like growth factor axis, the target of rapamycin, adenosine 5' monophosphate, activated protein kinase, and certain proteins. Present challenges in the aging studies are we have identified the role of many factors that promote aging, such as nutrient sensing metabolic pathways, loss of protein homeostasis, then increased genome instability, changes in the epigenetic marks, alterations in telomere length, etc. But still, despite these advancements, what are the individual genetic factors that are contributing to the aging and longevity is not yet fully determined. And how does the sex and environment affect in the aging process and the lifespan determination is not fully uncovered. The objective of the current research article that I'm presenting was to identify genetic regulators of longevity in a total of 3,276 UMHET3 mice. These UMHET3 mice were used in the longevity studies by National Institute on Aging's ITP program, that is Interventions Testing Program. This program was designed to find out what are the different interventions that can affect the longevity in the mice that were being studied. And second objective was to study whether the genetic basis of longevity is sex and age dependent. One of the findings is that females live longer than male. So is there any effect of sex of the organism in the determination of longevity of that organism? Is there any genetic basis for this? Then another objective was to study whether non-genetic factors such as litter size of the mice and effect of early access to nutrients on the growth contribute to the longevity determination. Methodology. So this study followed multi-factorial methodology in which they carried out a multi-center study to find out drivers of aging that an unbiased genetic and molecular cross-specific approach would reveal. Then the study started with genetic mapping of loci that modulate variation in the lifespan. So these UMHET3 mice, which were present in the different centers across the USA, and these were generated by using a 4-way intercross. These mice, these were part of an intervention testing program in which there were two groups of mice. Some were control mice and somewhere in which intervention or some drug was given. So all of these mice, these were allowed to age completely, and in the different parameters were tested throughout their lifespan, and these parameters were included in the current study. This test, ITP, that is Interventions Testing Program, it was conducted to test the positive effects of dietary and drug interventions. In ITP intervention, a large and sex balanced subcohort of control animals and test animals, these were also aged, and their test and treated cases were analyzed for differences in their longevity. So this 4-way cross, it was carried out to generate a UMHET3 mice were of this type. So females that were resulting from the bulb C by J crossed with C57BL 6J mice were crossed with males that were outcome of the C3H and HEJ crossed with DBA 2J F1 males. So this slide gives us an idea of the entire structure of the research. So this 4-way cross, the males and the females and males were crossed to generate the UMHET3 population. And this UMHET3 population had a different genotypes. The genotypes were further analyzed in sex and age specific longevity, QTL identification, QTL being quantitative trait loci, and relation between the early access to nutrition, growth, and longevity, and then age and genetically driven gene expression differences also. And these throughout this lifespan, different parameters such as body weight, litter size, and liver RNA expression, these were identified at different stages of time points in the life of the mice, and their effect on the longevity of the mice were estimated. So in the section age-specific longevity QTL identification, they found a particular chromosome on a locus on chromosome 12, which was found when both male and female were included in the analysis. When only female were included in analysis, a specific chromosome on locus on chromosome 3 was identified to be significant loci. And then after these loci were identified at that particular locus, what were the genes involved in the longevity? To determine that, they further analyzed the gene expression in the liver and also QTL gene characterization and prioritization by using different databases that are available, longevity-related databases, and also the UMH3 variants and their expression. So then they also carried out the analysis in CA11s to find the functional significance of those particular shortlisted genes, and they have also created a reference for further research. So here this slide shows the 4-way cross and different genotypes that were analyzed, and the parameters that were found from 3,276 mice, both in the control and treated groups. Then they carried out the QTL mapping, and they found out the significance of those particular genes in multiple species and came up with the candidate longevity genes. Here, this graph shows the longevity of lifespan of the mice. As we can see, females lived generally longer than the males. This is a QTL mapping, and here we can see that is pink. It shows statistical significance at chromosome 3, but there is no specific significant peak for any chromosomal location for males. But we can see a peak at chromosome 12 when both males and females were included in the analysis. Here we can see that to identify whether the QTL scans for lifespan performed, whether there was any effect when we exclude the early dates of the animals. So they have performed this analysis. So this x-axis represents how much of the mice population was excluded that were dying early. So as you can see, the longevity is increased here. And if we reduce those particular populations, given as 20%, 40%, 60%, 80%, many different loci increased in statistical significance in males, but not in females. But the chromosome 3 locus was found to be robust even to the exclusion of different levels of early deaths. And here, these chromosome 2, chromosome 10, chromosome 3, chromosome 12, and chromosome 12, both in males and females, these were analyzed to find out the contribution of different genotypes, BH, BD, CH, and CD, whether exclusion of early dates will cause any effect in increase in the longevity of those organisms, and robust statistical relationship between those particular locus and the longevity. Here, these different organisms were included in different longevity groups, and these are colored by these particular different color codes. So direct inverse correlation was found in males when in the earlier lifetimes, but in the later lifetimes, that inverse correlation was not there. So inverse relationship between body weight and longevity is stronger in males than in females at younger ages. So body weight, if there is an increased body weight, that leads to lesser lifespan as compared to the less body weight. Now they performed mixed effect Cox model hazard ratio analysis for per 10 gram increase in body weight in males and females, and they found that there is a negative effect of increased body weight to the longevity, and there is a direct effect of weight on the longevity. On the second level, whether the litter size affects the longevity. So they found that if there is an increased litter size, that means number of animals getting birth is more, then individual organism will have lesser body weight, birth weight, and that will have a direct effect on the longevity. So larger litter size, which leads to the lesser weight, will lead to the longer lifespans of the organism. Now here they studied the interplay between the early growth size and longevity of in the mice and also in humans to correlate the findings from mice into the humans. So this is a Mendelian randomization studies results. So MRIVW, that is inverse variance weighted Mendelian randomizations. And here they found the correlation between the early body size, early height, BMI, and height on the longevity of the organisms. So they have found there is a relationship between early body size and longevity. Then early height had a direct effect on the longevity, and this is a complex relationship in which the height had an indirect effect on longevity because there are some social issues involved here also. So a direct inference cannot be made because height is also indicative of social stature, and that will lead to the better opportunities, and the individual might get better income and will lead good life as compared to less height individuals. So that is one of the factors. So MR mediation, it showed that the effect of early height is not mediated by the height. So early height, that is the height in the younger years, it is not directly affecting the longevity. Then they also found effect of the early body size on the longevity. So the direct effect, there is direct effect on longevity, and the indirect effect is mediated by BMI. So median analysis, Mendelian randomization mediation analysis, it showed that the effect of early body weight is mediated by BMI. Then adult and early height, they showed genetic associations. So MR was performed with the IVs that are specific to each phenotype and excluding the common variations. So further enhancing the opposing effects of the early and adult height. Then MR mediation shows that the effect of early height is not related to the BMI. So here they have performed these analysis to find out the interplay between the early growth, size, and longevity in mice and also in humans. Then QTL mapping of the body weight, it identified sex-specific loci and evidence of shared genetic effects on the longevity in males. Here they have identified significant loci which are indicated in red, and these were specifically associated with the longevity. Then they identified further, carried out those particular chromosome 9 and chromosome 10 analysis, and found out what is the effect of genotype on the longevity. And further, they carried out mixed effect Cox model for the association of genotyping and body weight as a function of different truncation proportions. Whether there is a effect, age-related effect on the longevity by excluding the early death, that was carried out. So whatever the red arrows are shown, these were further analyzed to identify the candidate gene loci. Here they have performed the liver gene expression on those particular genes in loci that were identified to be statistically significant, to identify which are the specific genes that might have a play in the determination of longevity. So here they created a scoring pipeline in which they prioritize the candidate genes. So whatever QTL mapping results were identified in that, in UMHET3 mice, if there are high impact variants in the parental strains, then they will get a point. And then again, if there is a differential expression with respect to age and haplotype specific expression in the is present, then they will get additional points. Then these are the different databases. So if those particular genes are previously present in those particular databases, they will get an additional point. And in this way, they identified the top scoring candidate genes, and those top scoring candidates were analyzed in C. elegans RNA and longevity experiments to verify whether those particular genes had any effect on longevity or not. So here, these are the different chromosomes and genes that were analyzed in these studies. So female longevity, male longevity, and different levels of truncation, 0 truncation or 20% truncation and 80% truncation. So these are the different scores that these genes received, and these are the final genes HPK1, then DDoAST, UNC52, in humans HSPG2, FGD6, and PDK1. These different genes were analyzed for their correlation with the longevity. And they did indeed find statistically significant relationship between these genes expression and their effect on the longevity of the organism. So discussion, the effects of genetic factors are mostly sex, age, and trait specific. Many of the QTLs seem to modulate lifespan only in mice beyond a certain age. This was shown by the truncation analysis, and this is more prominent, especially in the males. The genetic determinants of longevity and body weight variation are different and sex specific, even though the two traits are highly correlated. The segregating alleles that do influence lifespan in UM-HET3 mice may be effective in one, but not in both sexes, leading to longevity differences between the sexes. Non-genetic factors such as very early growth and access to nutrients also play a role in determining longevity. Limitations of this study. It is possible that the lifespan is modulated by a large number of loci with smaller individual effects, so what is called as a multigenic effects. All of these loci were not studied in the present research. So there are only few loci which were found to be correlated with the longevity in the mice. So only those loci were studied in this particular research. The study was conducted on UM-HET3 population. Now this lab-grown mice is inbred for hundreds of generations, and therefore selection for breeding success in laboratory conditions, and therefore they may have lost the genetic variants present in natural mouse populations. Not all aspects of mouse physiology are expected to be identical in humans. This relates to the whether we can translate the findings from this present study for the human genetics relation to the longevity. Conclusion. Now, this study results provided insights into determinants of longevity, highlighted genetic mediators that can be sex or age specific, and highlighted non-genetic effects such as early access to nutrients. The output provided by this study will be useful for the hypothesis building for future longevity studies and devising therapies for aging, age-related diseases, and longevity. Impact of this research, the characterization of genetic and non-genetic determinants for longevity in mouse model will be helpful to explore the determinants in humans. Further studies at the population level may identify genes and pathways involved in aging. The research extends the reach for future possibilities for targeted anti-aging therapies and extension of healthy longevity. Thank you for patient listening.
Thanks, Vijay, for the nice talk. So the forum is open for questions. Okay, I have a question, that is, what will be the impact of this research on the human longevity study? That means that they identified some genes for the mice, but will this is applicable to the human also?
Yeah. So this is an important question and also included in the limitations of the present study. We cannot perform this kind of study in humans directly, because that will not be ethically right. To grow humans specifically in a controlled conditions just to identify their longevity is not ethical. So we need to have some organism model to study this in, and mice are the good ones, and they do share very much the genetic factors. Most of the disease studies have contributed in mice, has contributed to the human research also. So what present study does is that they identify that there is indeed a sex-specific difference in the longevity, and they have identified two chromosomal locations, chromosome 3 and chromosome 12, and they have carried out, they didn't stop just at identifying with the statistical correlation. They also performed laboratory studies in another different organisms also, here being C. elegans. So what this study will do is that if there are certain genes which are corresponding into the humans, orthologs in the human, now those can be studied in larger populational studies, because more and more genetic data at a population level is being unraveled in different studies in which we are going for genome sequencing of the many human populations. So in future, whatever candidate genes that will be generated from this study, they can be tested in humans also, whether they are indeed contributing to the human longevity.
Okay, yeah. Thank you for the nice explanation.
Thank you.
We welcome you in our next presentation. It will be presented by Jay Darji. He will present on a paper titled Deep Match, a methylation aging clock developed with deep learning. We wish to meet you in our next presentation. Thank you.